Since the 1940s, many antibiotics have been discovered and isolated to fight bacterial infections. However, over the past 40 years, there has been a major decrease in the number of antibiotics being produced. Additionally, there has been a global outbreak of antibiotic-resistant infections.
To address this issue, Yale launched the Small World Initiative with the intent to discover new antibiotics to combat the growing threat of antibacterial resistance. Through the Small World Initiative, undergrad students from around the country will be isolating bacteria taken from soil samples in order to discover and cultivate novel antibiotics.
So why soil? For three reasons. First, there is an abundance of microbes found in the soil, making for easy access. Second, antibiotics were first discovered in the soil. And third, it is cheaper to obtain isolates from the soil rather than having a team of scientists produce synthetic antibiotics. As a part of the Small World Initiative, each member of our group went out and collected soil samples from different areas. I collected mine on Bethel campus behind the Upper Heritage Lot in a densely populated deciduous area. I chose this area because I thought the decaying organic matter of trees would support a high microbe population.
While Max collected his soil sample from a deciduous area on Bethel's campus, I collected my soil sample near my house, from an area surrounded by several coniferous trees. I chose this location because I was curious to see how the microbe population would be affected by the environment at the foot of pine trees.
I, like Max, obtained my soil sample from a deciduous area on Bethel's campus. I went as far into the woods as Bethel Wi-Fi would allow so that I could record the GPS location. After clearing away the snow, I used my digging utensil, a plastic fork, to scrape the top layer of frozen soil into my container, which I brought back into the lab. There, our team collectively diluted our own samples to single out individual colonies and obtain the CFU per gram and Shannon-Weiner index. This is a picture of my beautiful microbes, taken on Valentine's Day. As you can see, the plate in the front has the highest concentration of colonies, and the concentration decreases as we proceed to the back.
Microbiologists refer to single colonies of bacteria growing on a plate as colony-forming units, or CFUs. This table shows the number of CFUs per gram of soil for the three locations. Notice that location 3, the soil obtained from the coniferous area, has the lowest CFU per gram count.
The Shannon-Weiner index is a measure of diversity within a population of microbes. A soil sample with a higher Shannon-Weiner index would have a more evenly distributed population than a sample with a dominant microbe and other small populations. Again, the coniferous area has the lowest index. Our claim is that soil isolates from deciduous areas correlates with high viability, diversity, and antibiotic production. This is because soil samples from locations 1 and 2 were both obtained from deciduous areas. Both displayed a high CFU per gram, Shannon-Weiner, and antibiotic productivity. This contrasts with the soil sample from location 3, which was obtained from a coniferous area and has lower counts in all categories. We contribute this correlation to the lower pH of the soil at the base of the coniferous trees due to the acidity of the needles dropped from the conifers. These results are similar to Fryer and Jackson's claim, who are cited in the Small World Initiative Lab Manual.
Here are our master plates, and the tester strains that we plated our soil isolates against. Max plated 22 isolates, and had 1 hit on P. vulgaris. I plated 9 isolates, and had 1 hit on B. subtilis and P. vulgaris. Ryan plated 23 isolates, and had 1 hit on M. luteus.
Pictured here are the isolates, which we deemed as hits because of the zones of inhibition. A zone of inhibition is the absence of microbial growth in the vicinity of an isolate due to the presence of an antibiotic secretion. Because of a zone's temporary nature, the picture of Ryan's isolate, which was taken at its peak of antibiotic production, exhibits a larger zone than mine or Carle's.
Here, I am looking at the first plate we used to culture in mass quantities, the isolates that we have identified as hits. This was done so that we could maintain a viable source and isolate its secretions to identify its antibiotics. From this plate, we have selected our microbes to apply to slides, gram stain, and examine via microscopy.
When analyzing bacteria to determine its genus, the first step is to identify its shape. The most common shapes are coccus and bacillus. Cocci appear as spheres under the microscope, whereas bacilli resemble rods. Other less common shapes are spirochete, which appear as alpha helices, and streptococci, which are long chains of cocci.
When looking at each of our isolates, we discovered that they were shockingly similar in appearance. Everyone in our group had gram-positive rods with gram-negative clutter in the background. Before we could see these features, we had to execute the gram staining procedure. First, we heat affixed the sample onto the slide and then flooded it with crystal violet. The crystal violet is retained by the thick peptidoglycan layer of gram-positive cells, but is washed away in the gram-negative cells, as the thin peptidoglycan layer is located between the plasma membranes. Following this, we flooded the sample with safranin, which acts as a counterstain which adheres to the gram-negative cells, making them identifiable.
The crystal violet, as its name implies, stains the gram-positive cells a purple color, whereas safranin stains the gram-negative cells pink. Shown here is a close-up of Max's slide, demonstrating the difference between the gram-positive rods in the foreground and the background cluttered by gram-negative schmutz. From this we have determined that the significant cells are the gram-positive bacilli in the foreground.
This leads to our second claim that the gram-positive bacilli shown on the slide are active antibiotic producers in all 4 of the samples in our group. For all of these, the genus is Bacillus. We chose this because Bacillus is a gram-positive rod that is statistically highly prevalent according to the genus flowchart. However, they may be different species as they responded to different tester strains.